machine. So the question came up last week, why don't we just inject telomere? And wraps around like a paperclip. If it's an A here, it's a T here. If it's a G here, it's a C there, and vice versa. So what nature has done, and this holds true for all plants, animals, fungi, is to protect these linear chromosomes. The blue strand wraps around like a paperclip and then tucks back in to mate with the red strand. It's kind of clever. And on a ultra-structural level, we see that there's actual binding proteins that mediate this, and there's other sheltering molecules. None of this is important. You just got to remember the paperclip. And last week we discussed why telomerase activation is not carcinogenic, and here we have absolute must principle here. So this bow tie with the squiggles, double helix, is the chromosome. Here's the overhanging leading strand, that thing we just looked at, that's getting actively lengthened like an old Dymo label maker for your locker or your PG notebook. This kind of gets longer and longer if it's a stem cell. This thing is a bunch of proteins, and it's a machine. So the question came up last week, why don't we just inject telomerase? Well, telomerase is a machine. It's assembled within that cell for use in that cell. It can't cross into cells. And it needs to be synthesized. Each one of these proteins needs to be synthesized, and it needs to be assembled into a little, like Lego, micro machine. So it's only for use within that cell, and only for use within that cell's nucleus to repair that cell's DNA. The green is the RNA component, T-E-R-C, RNA component. That is the template, which is the printing press. It tells you which 6 base pairs are you going to print out in the case of humans. Maybe it would be 23 in the case of yeast. So this green thing is the RNA component, and this is the key to run the engine, which is the T-E-R-T, reverse transcriptase. The naming is somewhat troublesome, but RT basically is an engine. It's a mixture of a bunch of molecules. And RC is the RNA component, which is a single molecule, single RNA molecule. And this is the end that gets spliced. As we discussed last week, stem cells have a splice key, which works. It runs the keyhole, which changes the conformation so that the whole thing can start working. In a regular cell that's not immortal, the key is too long. It doesn't fit the keyhole, which is the case with that premature aging Discaron syndrome. And so the key is useless. And so in most of our cells, the key is useless. It doesn't matter if you have a lot of engine. You're not going to be able to write more DNA. Brief review of genetics. There's a guy named Gregor Mendel. He's a monk with some time on his hands. And he started breeding beans. And he found that, as humans have done for thousands of years in terms of husbandry of animals and plants, that if you cross certain plants, you get mixed traits. So if you have a green bean and a green bean, there's the green gene and maybe the not so green gene. So the children of this plant would be green, 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 because you only need one copy of the gene to be green. But if you had a not so green gene here and a not so green gene here, then 1 out of 4 times you'd get a white bean. Okay? And this holds true with people as well. So in a typical situation, let's say there are many diseases where the dad would be normal, 2 capital R's. The mom might be normal, but with a hidden gene with a small r. So in this case, if you do the squares, you're basically talking about there's 2 chromosomes, one from mom, one from dad. Chromosomes like this, and genes are just little sequences of DNA. So what Mendel was finding out was that you get 1 copy of a gene from mom, 1 copy from dad. So in this case, you can get 2 capital R's, totally healthy, 2 capital R's, double homozygous for the wild type or the healthy natural state. And you can also get a situation where half the kids are carriers because mom gives half her genes here, half her genes here. Kind of confusing, but it'll become picture of Yalta just before the end of World War II, and it just shows some heavyweights here. Got Churchill, we got FDR, and Joe Stalin here. And so they kind of decide how they're going to carve up the world there. There's three people here. Blasco, the lead author, Grider, who worked with Blackburn, the Nobel Prize winner, and also DePino, who's now at Harvard Dana-Farber Institute, who published the article on the mice with telomerase activation. So these people, 1997, were all present at Cold Springs Harbor and learning about how to engineer mice. So what did they do? They took these mice and they knocked out their Turk. Remember, reverse transcriptase is the whole enzyme engine, but RC is just that green key. So they created mice that were deficient. Mom and dad gave them a deficient copy, whatever. They have no ability to make a good key. So they found out that these mice, as they went through generations, got older. The G1 was the first generation of knockouts. The second generation were born older, and with all the damage. The third generation were even worse, and the fourth generation were not even viable. So, unlike the song from Bye Bye Birdie, What's the Matter with Kids These Days, that's the thought you need to think about, because look at Larry King over here enjoying his wonderful children. And the point is that Larry King, his sperm, his germ cells are immortal, and they're darn good. There's good error correction. There's infinite telomerase activity, protected caps. So what they did was to take these mice and make them telomerase incompetent by removing the key. So every generation was worse. You know, we only have the perception as we get older that there's something wrong with kids, but in actuality they're just as good.
Why can't they be like we were, perfect in every way? What's the matter with kids today?
And in this case, the kids of the mice were progressively older and worse. That's kind of a mean thing to do to mice, but it proved a point and built the next 14 years of research. So here is what that would look like. In this case, look at, let's say the mom is a double knockout mouse, one of these genetically mutated G1 or second generation, no telomerase, getting older mice. You introduce a half normal dad, and you make them, and you get, again, knockout. Half the kids are knockout, still not able to grow their telomeres. Half now have recovered one gene from the half-healthy dad. These are restored, okay, knock-in mice. So what they discovered, what Blasco discovered long ago, or maybe 4 years ago, was that if you took the bad mice and knocked it back in the third generation, well, look what happens. The normal telomeres are still long, the broken telomeres are still short, but the repaired genes, repaired. Knock in, long telomeres, and good news. Now we're switching gears to TERT. Again, that is confusing. Forget the TE part. Just look at the RT part, reverse transcriptase. That's the engine. So what Blasco did, and her resume just is a very interesting resume, because she basically asks big questions, fundamental questions, and tests them. And her career alone, in my opinion, has advanced the understanding of this field more than any other single investigator. And she remains a close collaborator with Jaron and with TA Sciences. And in fact, was featured last month in a news report picked up by the mainstream media regarding her test for critically short telomeres, which is commercially available in Europe. And the test was being touted as a test to tell you how long you're going to live, which is really not the case at all. But they did develop a novel test for measuring short telomeres way back in 2007, and now it's going to be available for people. So the reverse transcriptase enhanced mice, she took these mice that were enhanced in terms of their tumor suppression, P53, P16, P19. These are all the monitoring and the proofreading and the killing off of the bad genetically damaged cells. These were all very meaning that cells that probably wouldn't have had that were differentiated, she gave them that ability. And guess what? They lived longer and healthier, and they lived 40% longer. So it's the combination of having telomerase activity and previously telomerase inactive cells and the high level of error correction that made them all good. There was no increased cancer, and the lifespan was increased by 40%. Again, reverse transcriptase, we're not talking about the key anymore, we're talking about the engine. The Harvard mouse study that got a lot of press back in November and December of last year was by DePino, one of those guys that was present at Cold Spring. I think he came through, I want to say Stanford, UCSF, and now Dana-Farber a year or two ago. So he's made a great study here of what happens when you take these engineered mice. These are not knockout Turk mice, okay? Not like the ones they created in '97. This is knockout for Turk. Something about the reverse transcriptase is defective here in both copies from mom and dad. So they made these mice that age fast, but they included a lock and key mechanism, meaning they could turn it on at will. It was like a latent gene. So they created these mice with a lock, and the key was a tamoxifen derivative or a relative. Not important. It's just a steroid hormone that could activate the telomerase. And what they found, to their surprise, was that the mice got younger, like Benjamin Button. Not only did they not get older, which they were anticipating, just an arrest of aging, but they actually saw youthening, if we can coin that word. The skin, the pelts got darker. The neurological system measured by, smell and all these other things got younger. And so this was exciting. People said, well, that's neat. Wouldn't that be great if we could do that with people? Hello, we've been doing that with people since 2007.
You're listening to this?
That's exactly what TH65 does. And that's why I believe, and my patients are...
The things they can do with this genetics is unbelievable.
Showing that people are getting...
Now, they got these mice, they got one generation, then they altered the gene, and then the next generation was born old. Next generation was born old again, and worse. Next generation was unviable. And they go and do another one. It's time to do the reverse that. And it gets down to a super mouse that lives 40% longer from an old mouse. It reverses the old mouse back to a young mouse.
So that movie about Donald Button, what was his name? Button.
Yeah. So they've done it with mice, and there's no problem doing it with humans, apparently. Telomeres.
So how does TH65 work? Well, how do we know telomerase is active? There's something called TRAP, which is telomerase reverse transcriptase activity protocol, I think. It's something like that. It just means you put it in a dish and you see if there's more activity. And the way you know that it's active is that new base pairs are added to the telomeres. This is standard measurement that people do to measure activity. So if you want to know whether yoga or good exercise or sleep increases telomerase activity, you would use a TRAP assay. So the paper that came out in April by Blasco with the TA Sciences folks and Cal Harley, who's the lead scientist and discoverer of TA65 from Geron, showed that at 24 hours at this concentration, TA65 increased telomerase activity in these third-generation mice. So now we know, remember, that G3 are the ones that their parents were incompetent, but now, being spliced with a half-normal dad, they have some telomerase activity. And if you could read this, I apologize for the size, it would show positive and negative. It's third-generation mice, and this high bar is showing that they have 2.5 times the telomerase activity. And that's good. Here's the double negative, G3 without the crossing with the normal, half-normal dad. There's just no activity there in the telomerase. And the interesting thing is that also this result did not persist at 5 days. So it's a transient effect. Translates translates to those of you taking TA65, it's not going to be there 5 days later. Okay, it's only, for the 24 hours that you're taking it. And I think people who go on and off can attest to that. There is a certain immediacy. So what happened in 2011 April was she published these articles on the mice, but she did this just to dot the I's and cross the T's. She already knew what the result would be because she did this experiment in 2007 where she had the Turk knock-in mice, and she found that these histograms of the end-to-end fusions that we've talked about in the past, you went from this number to none. Okay, it's clearing these bad double chromosomes. End-to-end fusions to none. Fragments, breaks, still present, but these rings, they went to none. So something about making telomerase active by restoring the key allowed the cell to go through its final apoptosis, or killing off of those damaged chromosomes. So fast forward to 2011, the recent study, she found the same thing here, that signal-free number dropped when you added TA65. Critically short telomeres dropped, adding TA65. And this is something which is a signal of DNA damage, which would occur if you had, say, oxidative damage or just an uncapped telomere. There's a signal, and this shows that that signal was decreased greatly when you're taking TA65. Now, the good news is that although the effects of reverse transcriptase activity are only dependent upon the time that you're taking it, the repair is permanent. And so we know that because if you look here 2 years out, and this is 3 months after treatment, the control group still had a lot of short telomeres, had broken telomeres, although the treated group, even long after this stuff was out of their system, the T65 group had less. So what's going on there? The only way to explain this is that, in my opinion, the damaged stem cells, the damaged queen bees, if you will, were kicked out and they were replaced by younger ones. And the copies of the younger cells, this is 3 months after treatment, they're making better copies of themselves. And that will come into play next week when we talk about weight, diet, and obesity, and how I lost 25 pounds, even, and it's impossible for me to put it back on. It's because the same cells are not present. They're less damaged now. They're healthier fat cells. So the point is that aging is an escalator that's going down, and when you take good care, you're able to stand still. If you don't take good care, the speed of the elevator catches up and you start to descend. But if you exercise, eat well, sleep well, reduce your stress, you run up the escalator, and then you are younger physically. It's kind of like pouring champagne into a tower of champagne glasses. And this is a big issue with people who maybe will notice an immediate effect, and then they'll say, well, I don't feel anything now. Well, you know what? You're trying to fill the glasses down here. You still have some damage that needs to get taken care of. And so you don't get old overnight. You don't get young overnight either. And that's the thing that a lot of people, dependent upon their psychic makeup, find difficult about taking TA65. They feel great initially. Maybe some arthritis goes away. Maybe they lose some weight, but they're like, I don't feel it. I just don't feel it. Some of the people with the most damage, I remember Chuck was one patient who had really horrible sleep. That sleep improvement didn't kick in until about 6 or 8 weeks because there was so much more in these champagne glasses that needed to be fixed. And so every night, you only have so many things you can fix, and you wake up and you go at it again, damage. And so it's a cumulative, gradual process of repair. But interestingly, although there are mechanisms to repair DNA,
they're doing it already,
which we can get into someday. Probably the main way TA65 is working is by allowing them to be destroyed, break the champagne glasses that aren't working, and recruit ones that are going to be younger and healthier. And we discussed that in it's called the mesenchymal stem cell recruitment, and that's a different podcast, which I'll refer to in the So that's it. short one today. If anybody has questions,
Yeah, they were talking about
I'd be
evil.
happy to entertain those.
Because obviously they've got it.
Question. Kensmeyer. It seems that TA65 is really better.
Still dependent upon the soul.
That's what I'm saying.
I don't know. Does it seem that way? I know people who've gone off it after starting TA65, and I know people who remain on it. I think they're different, but perhaps complementary. Of course, HGH can be overdone. It can be supercharging, so I'm not a big fan of that. Ralph asks, with the replacements from the better stem cells, does the number of stem cells increase again? Think of it like this. You only have a certain number of queen bees per hive. So if there's a queen there and she is damaged, she'll often go into a state of, she can't copy because the DNA watchman, the P53, is keeping her from copying, but she's still doing her job. She's kind of phoning it in. She's kind of like a 63-year-old middle management person in state government. She just wants to phone it in. She's not really at 100%. So what happens is, and they don't promote from within. The drones usually don't de-differentiate, although that happens in muscle cells and satellite cells, as we discussed in podcast 11. What happens is there's a vacancy, and then these itinerant stem cells, these mesenchymal stem cells that are ready to go, they're college educated, they're smart, they can do this job, that, or the other. They come in and become the new queen bee in whatever niche that is. So anyway, it's a concept of stoichiometry. Somehow cells signal each other to know that there's enough people here. We don't need anybody new, no new guys, and we already got a boss. Okay, and so what you need to do is kill off the bad cells in that niche. Usually, there's only one less differentiated stem cell that replenishes all the drones around her with her bad or old DNA. So I hope that kind of answers your question. Okay. David from Manhattan Beach asks, what about menopause? Does the loss of estrogen create special problems for women? Yes, it does. Unlike andropause, which is a made-up thing, not recognized by the World Health Organization, women about the age of 50 run into a wall called menopause, and their ovaries really stop functioning to produce hormones. They still have hormones from the adrenal gland, which can be converted into all the different steroid hormones, such as testosterone, estrogen. But it drops pretty much precipitously. The upshot of which is decreased bone density, decreased energy, sometimes the mood, especially during the transition, is poor. Hair quality, skin, nail, hair, especially in the sensitive tissues below the waist, it can lead to incontinence and dryness, etc. So Kent is on, but we only hear his radio. Yeah. Okay. Question from Kenneth. In the TA65 supplement, do you continue to take it for life, or is there a specific time frame that you take it and then stop? Excellent question. Well, as we discussed here, the repair is permanent. So these mice were treated, weren't taking it anymore, so the trap assay was no different. They weren't getting any benefit. And yet, you measure their telomeres, they continue to have fewer short, critically damaged telomeres. So the question is, do you continue to take it? Well, I would, and I do plan to, because as long as you're still using the car, driving it, taking it out, it's still getting old. So why not reverse or, stop that aging process? It's a matter of finances, but, 20 years from now, I'm sure Monsanto will have engineered corn to produce high yields of this substance. And, it'll just be a political issue, whether people will be entitled to stay young. It raises huge social questions. Although, in reality, the Medicare problem would would be solved, but you'd have a lot of unemployed doctors and insurance salesmen. So anyway, this is way above my pay grade, but certainly whether they're deserving or not, most people could benefit from a healthier lifestyle while they're here, and definitely something that I would consider taking, definitely. Rust never sleeps, as Neil once said, so you're always rusting, you're always oxidizing, and you're always aging, so why not continue?
Let me get something, let the screen recorder going so it can...
Don Wright, if I cut myself shaving, will I heal?
Still recorded?
I don't think so.
Oh, wait on. I'll take this out so Yah can hear too.